started doing research, working on the development of technology to genetically modify plant cells. I was fortunate to participate in the group that actually developed the technology to produce transgenic cells and transgenic plants. And I went back to Mexico and I started working on two main topics. One was on sequencing the genome of crops that are native to Mexico, like corn, chili peppers, beans. And the second topic was studying the molecular mechanism by which plants adapt to environmental stress, particularly drought and nutritional stress. So I joined Texas Tech in 2018, and my research now is focused on trying to figure out the mechanisms and genes that are responsible for plants to cope with abiotic stress, but now extended to drought, salinity, heat, and trying to find the genes that could be useful to improve the tolerance of crop plants to abiotic stress. I discuss with my students and collaborators the hypotheses we want to address, the questions we want to answer, and then I give them complete freedom to develop their own projects. So I guide them, I try to keep them on track of what they do, but they have absolute freedom to develop strategies, to find new ways to answer the question in a faster, more efficient or cheaper way. When you have established collaborators, we have more fixed points of view of what to do. But when you discuss with students, they have a fresher mind. They really want to explore questions that might not be so obvious. So that's the part that I really have enjoyed the most in all my career, working with the students and seeing how they grow and how they start to be creative and design their own experiments, and at the end they can design their own projects. So I think that's the most enjoyable part of research.